Welcome everyone on the next presentation of the ThalesNano webinar series. Today's topics will be about cleaner chemicals and biocatalytic hydrogenation in the H-Cube. Our guest speakers will be Holly Reeve and Sarah Cleary from HydRegen from the University of Oxford. They will talk about how HydRegen's biocatalytic hydrogenation technology will help you to harness selective biocatalysis in the H-Cube systems. As usual, you will have the opportunity to ask your questions after the presentation, which will be answered by Holly and Sarah. Please feel free to ask anything. Now let me hand you over to Holly.
Good afternoon, I'm Holly and welcome to the HydRegen ThalesNano webinar today. We're really excited to be here and to talk to you in a bit more detail about what we're doing. So HydRegen is a 2021 University of Oxford spin-out company, and our vision is to become a global go-to company providing sustainable solutions across the chemical sector. We want to achieve this by helping our customers to tackle energy and resource efficiency, to embed continuous flow processing all the way from early stage R&D through to commercial processing, and to generally help the sector move towards more circular and bio-based economies. So I am Holly Reeve, the CEO of the company, and later you will meet Sarah, who is our Chief Scientific Officer, and she will take you through more of the technical details of what we're doing. Today we want to talk to you about why biocatalysis is a useful tool, what our biocatalytic hydrogenation technology actually does, how and why it works so well in the H-Cube, and finally about how we can work with you going forwards. And I want to make a disclaimer at the start. I'm going to assume that some people listening today are already using biocatalysis and are already really familiar with it, but that others have limited or even no experience with biocatalysis at all. So why biocatalysis is probably a good place to start. I don't want to spend too much time here, but I do want to highlight that biocatalysis is already pretty well established, especially in the pharmaceutical sector, for the introduction of chiral centers. The first example I'm highlighting is really well documented. It's the manufacture of sitagliptin. So in this case, a metal step was replaced with a biocatalytic step for the introduction of this chiral amine, and ultimately that led to really, really high EE and a knock-on kind of benefit for cost and environment. And at present, I guess that's fairly typical that one or two steps in an entire synthetic pathway might rely on biocatalysis. However, the second example is a case where the synthetic route became entirely biocatalytic and relied on 9 different enzymes. This took the synthesis from the traditional synthesis of 12 to 18 steps all the way down to 3 steps. And other benefits included control over stereochemistry, less electricity due to the much milder operating conditions, use of aqueous solvent, far fewer reactors required, no purification steps required, and a higher final yield. And I guess this is a trend we really want to support and to see kind of happening more often. But for now, let's focus on hydrogenation reactions. That's what we're currently all about. And that's simply the addition of 2 hydrogen atoms into a molecule. This represents a really large class of reactions and typically results in the creation of a chiral center. So let's consider how this is currently done. I guess the state-of-the-art approach uses hydrogen gas as a really clean and efficient hydrogen atom source, and that's fantastic because there's very little waste. Increasingly supported metal catalysts can be used, and they are easily removed from solution at the end. The infrastructure for running these hydrogenation reactions in batch and flow mode is really well established, and that's both at the R&D scale and also at the kind of large commercial scale, and those protocols are really well tried and tested. However, these reactions usually operate under really intensive energy conditions, so high temperatures, high pressures, and achieving high selectivity can still be a real challenge. And of course, those metal catalysts are expensive, toxic, and often of finite supply. So the second method is the use of biocatalysis, nature's catalysts, and this gives unprecedented control over the structure of the chemical product, and that's leading to its widespread use in pharmaceutical chemistry and also in specialty chemistry for the creation of chiral centers. In contrast to metal catalysts, these reactions operate under very mild conditions, often in aqueous solution, although we are seeing increasingly good compatibility with organic solvents, and the catalysts themselves are renewable and non-toxic. But at present, I think it's fair to say that particularly for these biocatalytic redox reactions, there are no go-to off-the-shelf kind of processes and scale-up routes. And we often hear that that's a limitation for increasing adoption. We also hear from people that are not currently using biocatalysis that the barrier to entry for this type of process is just really high. You know, you have to think about cofactors and cofactor recycling, and that can be really off-putting and hard to get started with. So to understand a little bit more about this, I'm going to hand you over now to Sarah.
Thanks, Holly, and hello, everyone. My name is Sarah, and I'm the Chief Scientific Officer at HydRegen. I'm going to introduce HydRegen's technology, but before that, I'll give a little bit of background information for context, including some more detail on how biocatalysis works, and I'll discuss the current challenges associated with biocatalysis used for chemical manufacturing. I'll then discuss how the HydRegen technology offers solutions to these challenges through our use of biocatalytic hydrogenation for chemical manufacturing. When biocatalysis is used for double bond reductions, the enzyme that performs the reactions rely on sophisticated hydride sources. These hydride sources are biological cofactors like NADH. Now, this is in contrast to metal catalyzed hydrogenations, which simply use hydrogen gas as the reductant. The sophisticated cofactors, on the other hand, are used by enzymes, leading to high chemo, regio, and enantioselective double bond reductions. However, these cofactors are quite expensive chemicals. The NADH is therefore recycled in situ as a way to minimize its impact on the cost of the process. This allows a catalytic quantity of NADH to be used rather than stoichiometric quantities. The industry standard for regenerating the cofactor is through the use of a second enzyme, shown here in green. But enzymes that produce the NADH from NAD+ are powered by biological energy sources like glucose. This results in large quantities of carbon-based waste. The waste generated also leads to a change in pH. Therefore, the reaction pH must be monitored and adjusted over time in order to maintain enzyme activity. However, the use of enzymatic NADH recycling is essential because it is the most straightforward method that ensures that the hydride is inserted at the 4 position of the NAD+. Reduction at any other position on the NAD+ leads to biologically inactive cofactors, for example, at the 2 or the 6 position. And this renders them useless for use by the red enzyme for the double bond reduction process. So therefore, overall, it would be ideal to have a lower waste, lower cost alternative to the current enzymatic NAD reduction steps. With all of that in mind, we at HydRegen recognized that minimizing the amount of waste generated per mole of product is likely to aid in increasing the uptake of these exciting biological catalysts for chemical manufacturing, particularly for bulk chemical production, where the value of the product itself might actually be lower than the cost of the glucose reductant. Therefore, it would also be essential to find ways of reducing the cost of the biological reduction step. We also identified that it would be beneficial to develop new ways of enabling biocatalysis in continuous processes, both for large and small scale R&D. This would ideally be developed for existing infrastructure and equipment, thus avoiding the added expenses associated with equipment overhaul. So our HydRegen technology enables the switch of energy source to hydrogen gas, no longer relying on glucose. This means there is no waste generated from the NADH recycling, and the pH of the system does not change. Our catalyst is also heterogeneous, which enables simple catalyst recovery. The immobilized enzymes are also stable for longer reaction times and have enhanced storage life. Catalyst handling is akin to common metals on carbon, such as palladium on carbon, but where the beneficial properties of the enzymes are retained, meaning the unprecedented enzyme selectivity remains. We still are able to use the green mild reaction conditions seen with traditional biocatalysis, and reaction solutions are aqueous, but luckily with the ability to use organic co-solvents to assist with substrate solubility. Our patented HydRegen system is composed of 2 enzymes that work together to harness H2 gas for NAD generation, shown here in the green and the blue illustrations. A third enzyme, shown in red, is then selected, which completes the desired double bond reduction step. We have shown that this third enzyme can be used either co-immobilized with the two HydRegen enzymes or can be used in solution, offering some flexibility in overall reaction design. The third enzyme can be interchanged based on the desired double bond reduction. In later slides, I'll show some case examples of carbonyl, alkene, and amine reductions. So a customer could use a proprietary NADH-dependent enzyme or purchase one from a supplier. HydRegen can also source these third enzymes, and as part of a joint development project, we can use our expertise in the area to assist in the enzyme selection. Overall, our technology has been proven with up to full conversion and perfect enantioselectivity. And owing to our use of hydrogen gas, our process is atom economical with no pH change, and reaction workup is simplified thanks to the easy catalyst removal. We have proven our process in batch and excitingly see that with each step in the scale-up process, there's improved productivity. Our proof of concept work was completed in the microscale, after which we used parallel reactors to optimize the catalyst and reaction conditions. We have since intensified these optimized reactions using typical laboratory equipment, and again, with catalyst handling similar to that of palladium on carbon. So simple degassing of your reaction solution, followed by either attachment of a balloon to a round bottom flask or charging of the pressure vessel. And finally, the HydRegen catalyst has been demonstrated in scaled-down hydrogenation reactors. So here, the Biotage reactor mimics industrial-scale autoclave hydrogenation reactors. So we were really excited to find that reactions completed in the Biotage were comparable to those which we performed in our laboratory equipment. The implications of this are that HydRegen technology is readily used for chemical synthesis using standard reactors and standard hydrogenation procedures. Case examples of chiral alcohol production showed high conversions and enantioselectivities. We can use different varieties of enzymes to enable the selective formation of either the R or the S enantiomer, as seen with the top two examples. Also, no dehalogenation occurred with the substituted aromatic groups. So this overcomes over-reduction challenges that can often be seen when palladium on carbon is used. We've also demonstrated a more relevant preparation of the API fragment quinacridinol, so the middle compound in the bottom row. This was then taken on to complete the synthesis of the drug sulfasenacin. The quantitative quinacridinol isolation allowed us to avoid the need for a column purification and relied simply on the filtering off of the heterogeneous catalyst followed by basic extraction. Also, the perfect enantioselectivity allowed us to simplify the overall process, avoiding the need for recrystallization. Other API fragments have also been demonstrated in batch. The top two substrates here initially presented us with some solubility challenges in the aqueous solution, but gratifyingly, we found that simply by adjusting co-solvent gave satisfactory results. And while each example here still has some room for optimizing in order to achieve full conversion, neither of these products resulted in any side reaction, such as dehalogenation or reduction of the nitrile group. We've also applied the HydRegen technology toward the production of flavor and fragrance compounds. When we began with citral, we isolated the geraniol product exclusively. This is highly beneficial as it avoids the need for intensive separations of the two isomers, geraniol and nerol, which is notoriously difficult. And for reference, when sodium borohydride was used to reduce the citral, a mixture of the two alcohols were produced, therefore underlining how the use of the HydRegen system alongside carbonyl reductases can be exploited in order to simplify the product purification. Beyond carbonyl reductases, we can also use HydRegen with other NADH-dependent enzymes. Ene reductases used the NADH hydride to reduce activated alkene functional groups. So here we demonstrated that the ene reductase used with the HydRegen catalyst is selective for reducing the activated alkene functional groups, leaving behind the carbonyls and the non-activated alkenes. And then the cinnamaldehyde example, we could also use both ene reductase and carbonyl reductase in one pot reaction. And this resulted in the fully reduced functional groups shown all the way to the right. And this is exciting. This one pot procedure is exciting from the perspective that it aids in the overall process step economy. And finally, amino acid dehydrogenases and imine reductases are also NADH-dependent enzymes. And we have used them alongside our technology to produce chiral amine products. These diverse reactions were exciting demonstrations of the HydRegen technology for chemical synthesis in batch reactions. And I'll now hand over the presentation back to Holly, who will discuss the translation of our catalyst into continuous flow and the new opportunities that these are bringing about.
Thanks, Sarah. Hi again. I thought it might be time for a little recap. So biocatalysis is a really attractive tool, especially for generating chiral centers, but can be limited by the kind of available scale-up routes. As we have scaled up so far, we've taken advantage of standard hydrogenation protocols, and we've achieved better and better results as we've gone up the larger and larger kind of batch chemistry scales. This is likely due to improved hydrogen availability, even though we typically get our best results at 1 to 5 bar hydrogen. Our catalysts can be handled very similarly to carbon-supported metal catalysts, except that ours are tolerant to small molecules, so CO, CO2 and oxygen. We also don't need high temperatures or pressures, and actually we get our best results at kind of 1 to 5 bar hydrogen in room temperature. We observed a trend in moving hydrogenation reactions into continuous flow for improved safety, improved control, and I guess just moving towards continuous manufacturing and automation in the future. And we were really interested to see if this would work well for us. So learning from our batch chemistry journey, we wanted to try out an existing reactor, and we decided to turn to the H-Cube. We turned to the H-Cube because of, I guess, of the in situ hydrogen generation from water, offering a really safe way of using hydrogen with really easy pressure control, but also really easy temperature control. We really liked the idea that if you're using an H-Cube already with a metal carbon column, you can just switch that out and put in our biocatalyst column. And we really wanted to just prove that that was possible, that we could take out the hard work of trying biocatalysis. And I just want to show you that all of our results, kind of, well, at least all of our early stage academic results are already published in Frontiers in Chemical Engineering. So feel free to go have a look at what we've done so far. The kind of flagship result in that paper is here, and that's where we showed that our catalyst was really stable over a number of days. We had really high turnover numbers, good turnover frequencies. Ultimately, we made 0.5 gram of a very simple case study product at this point. But actually, as a company, we've since gone on to kind of exceed these results. So we've now gone on to show higher productivity, to do really good conversions in single pass H-Cube reactions. The goals we're really aiming for are achieving 1 gram of product in an overnight reaction, or a different mode of working where you can take one of our columns in and out of the reactor, store it in the fridge, and each time you put it in, do a different reaction and get about 100 milligrams of product over the course of a few hours. And one thing we'd love to know from you guys, if you're willing to get in touch, is if those really sound like good key operating metrics for our first product. So that's being able to make 1 gram of something overnight, or alternatively being able to use this column a number of times for different reactions, making about 100 milligrams each time. So how can we work with you? Well, we want to make it really clear that we want to work with you wherever you are on our biocatalysis adoption scale. And we also believe that we have something to offer you wherever you are on our scale. So if you have little or even no experience with biocatalysis, we are offering you a kind of complete solution. We're offering you a slot-in alternative, so a biocatalytic column that can be slotted into the H-Cube, or in fact to your batch or other flow hydrogenation equipment, enabling you to just get started with biocatalysis without having to really think about cofactor recycling, using your known, tried, tested methods. If you're really well established with biocatalysis, we want to offer you a new way of working with less waste, all the benefits of hydrogen-driven biocatalysis. We don't really need to know what it is that you're doing. We have ways that you can use your own proprietary enzyme for your key chemical step. We don't need to know anything about it. However, we would say that we have a lot of process and catalyst optimization know-how. So if you have something that's working quite well in your labs, there's a lot that we can do to help you modify the catalyst and really get it performing to its best if you'd like to take that to higher and higher scales. So how can we work with you in the near term? Well, we already have some columns that are ready to give out to people that just make NADH. So you will slot these into your H-Cube reactor, or as I already said, other batch or hydrogenation flow reactors. And you can use your own NADH dependent enzyme in there. We will kind of have protocols ready for you to either run these overnight to get a kind of gram amount of product, or to run it a few times, often even with a few different NADH dependent enzymes, to show a few different chemical steps, and they should have a good shelf life over kind of 1 to 3 weeks. And again, we're looking for feedback on those kinds of goals. We already operate these in batch mode, as I've said. We operate them as a slurry most commonly. We use different kinds of carbon for different reactor types, so you need to let us know how you wanted to run them for the best results. And these we typically just run under a balloon of hydrogen. That's how we get our best results. And again, we'd be looking to get those same kind of standards, so a gram of product in a reaction overnight, or 100 milligram of 3 to 4 products. And here we'd probably actually expect a much longer shelf life, because you wouldn't have to reuse those particles each time. In the midterm, we're really looking to offer a product that incorporates a ketone reductase. So basically a complete solution for doing alcohol production. This will have a broad scope alcohol, ketone reductase or carbonyl reductase, alcohol dehydrogenase. So it will be able to do a wide range of carbonyl reductions. It won't be able to do everything, but it will give you a kind of complete solution just to get started with. We will then, as Sarah said earlier, be able to offer you some joint development work to really help get the right enzymes for you to do whatever it is that you're interested in. I should also say we can't only do alcohol productions. NADH dependent enzymes do exist for all those other reactions that Sarah talked about, and we're really happy to get involved with those. We've already demonstrated them. And we're always on standby. We already have resources within the company. If you are interested in a process, you can always come and get in touch with us. And of course, ultimately, we would like to license this technology to you for your commercial processes. So really, we're offering you an R&D tool to try biocatalysis, or an R&D tool to evaluate biocatalytic processes. But we want to be with you on that journey all the way to a commercial process. And we are currently looking at running pilot scale demonstrations. So we really are going to have the data for you to show how this works at that large scale, to give you the reassurance needed that you can actually take this technology all the way through your R&D pipeline. So with that, I just want to finish up. I want to say thank you very much for joining us today. I was Holly, you met Sarah earlier. Please do get in touch and let us know what you feel about our products, whether they'd have been interest to you, whether you think we need slightly different goals for what our products can do. Of course, if you'd like to try one, we do have the capability to send out a couple of free prototypes for people if they're particularly interested to work with us on making sure that they're ready to sell. And of course, any other questions you have about anything we've said today, we'd be really happy to hear from you now, or of course via email. Thank you very much for listening.
Hi, thanks for joining us today. It was really great to be here. We've had a question about whether or not the slides will be made available, and we are really happy to make those slides available to you, preferably via email. So I know the webinar will be available on the ThalesNano website, but if you'd like a copy of the slides, please just drop us an email. Hopefully you've seen our email address. That's holly@hydrogenoxford.com. The next question we've had is about whether or not we're using metals or enzymes to do the cofactor recycling. So the system we're using is enzymes on carbon. We use an enzyme for hydrogen oxidation that is incredibly efficient at hydrogen oxidation. It's a hydrogen scavenger. So unlike metals, it kind of does hydrogen oxidation under really low hydrogen environments. It's also like oxygen, carbon monoxide, carbon dioxide, sulfides, and things like that. So that makes it a really good hydrogen oxidation option for us. And then we use a second enzyme, an NAD reductase enzyme, that does the biocatalytic NADH recycling, and that's how we achieve the really high selectivity for NADH generation. That's obviously really important to achieve. And those two enzymes are in communication with each other via a carbon bead. So yeah, at the moment this technology is metal free. If you have any other questions, please drop them into the chat box.
I'm seeing one that's asking, how are these biocatalysts handled and stored? And so we kind of mentioned during the recording that we handle them quite similarly to something like palladium on carbon. And specifically for the H-Cube, we've actually packaged them specifically into cat carts. That way they can literally just be slotted in, replacing a typical ThalesNano metal on carbon or metal supported metal catalyst. When we handle these catalysts in batch, they are typically in a slurry that can just be dumped into your reaction solution and then degassed and treated with hydrogen like you would with your palladium on carbon. And then in terms of the storage, it's more typical for biocatalysts where the preferred storage would be in a freezer. Our supported biocatalysts, because they're immobilized, you can do some shorter term storage in the refrigerator for, I would say, up to a day. But yeah, the preferred would be in the freezer. Any other questions? I'm seeing one that says, what is the lifetime of the HydRegen biocatalyst system? So what we've shown in some of our reactions is that you can get up to 1 million turnovers of the enzyme. So that's 1 million catalyst turnovers. And in terms of reaction time, we've shown reactions can run for multiple days at a time. So it's quite an exceptionally long biocatalyst lifetime relative to traditional biocatalysts that haven't been supported.
And there's a question there about the temperature and pressure limitations of these reactions. So we kind of work optimally at kind of room temperature to kind of 30 or 40 degrees, and we routinely work at pressures of 1 to 5 bar. We have actually demonstrated the system up to 50 bar hydrogen, and our catalysts are tolerant to high pressures to some extent. However, we don't see any advantage on going to those high pressures. As I said before, our hydrogen oxidation catalyst is a hydrogen scavenger, so it doesn't tend to be limited by the availability of hydrogen, but we do have the capability to go to those higher pressures. Our cofactor recycling system is also can be modified to be quite tolerant to the elevated temperatures, and we have the future possibilities of working kind of at 50 degrees and things like that. But yeah, we absolutely don't need to go to hundreds of bar hydrogen and hundreds of degrees, and that we would say is less a limitation and more a benefit of the technology.
Definitely. I'm seeing a question about what co-solvents we can use with this system and which ones need to be avoided. So far we've shown that both co-solvents that are soluble in water as well as those which aren't work well with the system. Actually, typically I first start out with a co-solvent that will be miscible with the water to see if that will help dissolve the insoluble organic substrate. And from there, if that's not quite doing the trick, then you can do a bilayer, so a biphasic batch or flow reaction where something like ethyl acetate or hexanes, and that's also proven to work quite nicely with our system. I would say in terms of avoiding co-solvents, it would just be any that might not be compatible with the third enzyme. So let's say you're using an alcohol dehydrogenase, you might want to avoid acetone because the alcohol dehydrogenase is going to also act on your co-solvent. But otherwise, so far our system has not, I haven't seen it be inhibited by any of the co-solvents other than something like that. Yeah.
I guess we could talk a little bit about how we see scale up going for these processes. So at the moment, obviously, we're talking about preparing our catalyst in cat carts for you to put into your H-Cube reactor for kind of gram scale production overnight. Sarah's already said that we can run these catalysts for many days, so that does open up the possibility of getting to multi-gram scale. But for kind of going to the higher and higher levels, we would definitely envisage providing some joint development work and then providing catalysts perhaps as a slurry for you to be able to put into proprietary flow or batch reactors. And if you want to go to even larger scales, then there's some other kind of models that we can envisage for that. But we certainly are tackling scale up of our actual catalysts. And we're working with some companies to look at kind of large scale processes so that we can give customers the reassurance that if they use our catalysts at small scale and they work well, they should follow through and you should be able to use them all the way up to commercial scale.
Great. And I guess if there are any other questions that come up after viewing the recording, feel free to get in touch with us at either of our provided emails or on our website, hydrogenoxford.com, and we'd be happy to be in touch.
Great, thanks for joining us today. It's been great to present to you, and we definitely hope to hear from you in the future.
Yes.
So.
Thanks, everyone.